Alright, so as we begin talking about epithelial tissue, what I'm going to do is, you should have already taken your notes on this. That's why you were supposed to look at the slideshow and then listen to the screencast. The point, your diagrams as you go through here and as you look at the slideshow, you want your diagrams to be as accurate as possible to what you've drawn. In other words, you're going to have to be able to look at these diagrams and know the tissue you're dealing with. So here's a few examples. You can see there's goblet cells, we have the cilia on top. It's very easy to see this is that. This is this. So you get an idea of exactly what's going on structure-wise. Now, that being said, a couple of reminders about epithelial tissues then are basic functions. So now as we look at epithelial tissue, recognize they're found in different places, and you're going to be looking at different functions associated with it. You need to recognize it's lining covering glandular tissue. In other words, there's always an exposed surface. Okay, so that's huge. Next thing to recognize is in the functions here, you need to be relating form to function. In other words, the different cells fit close together. We're going to look at how they're organized. Okay, you have flat, you have stratified, single layer, you have flat, cube, column-like. So which of those shapes, which of those layers are going to fit best for these different functions? And then recognize here they fit close together, lower surface always bound by a basement membrane. In other words, that's what links it. Since they have an exposed surface, that's what's going to link it down below. And to me, what's almost seems to be in spite of it is it's avascular. It has no blood supply. That means if I have my cells here, and here's my basement membrane, okay, that means all waste and nutrients has to get there by diffusion. Now, since all waste and nutrients have to get there by diffusion and they regenerate easily, that means even though they're avascular, they get a lot of blood, a lot of nutrients, the wastes are removed, they regenerate and duplicate, in other words, undergo mitosis readily and easily. Alright, so basic shapes again, flat, cubed, column-like, and they're either simple or stratified. So that means I can have simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar. You get the idea. Now, when you're looking here and you're doing these diagrams, what you want to note is you want your diagram to be as accurate as you can. So again, as I pointed out at the very beginning, you want to make sure you're drawing in all the detail you can see on this slide, because you're going to have to be able to recognize this on a microscope for the histology practical. Again, here, notice the two different layers. Here I'm looking at probably a transverse, okay? Here I'm looking at a superior or inferior view. In other words, this, okay, turned so that it's flat, just like that. Alright? Know where it's found. Let's deal with simple epithelium in terms of cuboidal. Now, notice cuboidal means square. These aren't quite perfectly square, but generally the shape is there. You can see the basement membrane below. We have a very pronounced nucleus, not the same as our simple squamous. Okay, we have a slight nucleus, but the cell layer is much, much smaller than these. That's kind of important in terms of the differences in function. Okay, so these have very different functions. Again, you can see the exposed surface, and you can see these small areas between. You can see that light area. That's what you should be drawing in. Okay, if we go to simple epithelium, a couple of points. Obviously, when we deal with columnar, simple epithelium columnar, we're looking at obvious columns. Now you can see this thickening up here. That thickening up there is actually the small fingers that we associate with microvilli. That increases surface area, okay, which allows a greater amount of diffusion, which is kind of important when we look at these. Also notice how packed these cells are, how tight they are with each other. Intertight junctions. Remember that we have the different junctions between cells. You have gap junctions, you have tight junctions, and you have desmosomes. When we talk epithelium, we almost always are dealing with tight junctions or desmosomes. Now, the other thing you want to note here is notice how most of these nuclei pretty much in the same plane. They're aligned with each other. You can see that here. Okay? Now, when we look at the next one, which is pseudostratified, okay? Pseudostratified, here's the cell, here's my column. Okay? Here's another column. Here's another column. They're also tightly packed, but notice here's a nucleus, here's a nucleus, 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 nucleus. See, the nucleus is not in a straight line, okay, like it was here. So that's what gives it that appearance of pseudo-stratification. In other words, false stratification, because the nuclei aren't in. Now this is a goblet cell. You saw that in one of the previous slides. Okay, so again, same thing, and notice also it's typically, we see it very commonly lined with cilia because it's going, we find it in the respiratory tract, so it's moving all of that mucus around. Now, the first stratified we'll look at is epithelium. Stratified epithelium we'll look at is stratified squamous. Here you can see the tile-like cells. Okay, I can see one here, I can see one here, I can see this one, I can see this one. See how they fit together? Okay, you can see the nucleus, 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 nucleus. You get the idea. So we see the layer. Notice that we see it literally growing from this bottom up. In other words, mitosis is taking place here. When we talk in the integumentary system, we'll talk a little bit about the keratinization process, but basically you see mitosis here, mitotic division sending it up, and we get these flattened cells. Okay, here another view, you can see the flattened cells and how they're all linked together. Now, stratified cuboidal, stratified columnar, big here, they're rare. You're not going to find them a lot. Notice here, if we look at stratified columnar, I can kind of make out the columnar issue of this. The one below looks like a cuboidal, but because of the columnar one on top, this is why I think it's stratified columnar. But since it's rare, we don't spend a lot of time on it. The last stratified we're going to look at is transitional epithelium. This dome shape here is indicative of this tissue. Okay, we haven't seen that dome look on anything else. So that last layer is dome looked, and then we have all of these kind of roundish layered cells as we go up. So that's the stratification. What's interesting about transitional epithelium is because of this dome shape, it gives it the ability, when it gets stretched, it literally flattens out and it looks like you're looking at stratified squamous, and that's why it's in the urinary system, right, within the bladder. Glandular epithelium. Big thing here is, since we're talking glands, that means we're going to be secreting something. So that means secretions. If you think about it, are we going to have a flattened tile-like cell, simple squamous? Is that going to be able to produce a secretion? So again, go back to form and function. Make sure your diagrams look as accurate as possible.